Hey guys, it's Audrey.
And Annie, and welcome back to our channel. Today we are going to be covering transcription. Transcription is the first part of protein synthesis, and it is the process of creating proteins from genetic code found in DNA.
For example, proteins such as keratin undergo transcription in their protein synthesis process. Keratin is commonly found in supplements that improve hair, skin, and nails.
The process of transcription involves DNA being transcribed or copied into a new complementary RNA strand. This is the main aspect and result of transcription. The purpose is to create this complementary RNA strand. Let's take a deeper dive into DNA. For example, here are the first 24 bases in a keratin DNA strand. DNA is a genetic blueprint for all living organisms, even us. DNA is made up of two strands, a coding strip and a non-coding strip, twisted together into a double helix. There are three components that make up DNA. First, the phosphate group, and then the deoxyribose group, which is a deoxyribose sugar and serve as the backbone of DNA. Then there are the nitrogenous bases, aka the nucleotides, thymine, cytosine, guanine, and adenine. These bases have very specific base pairing rules, as thymine, or T, pairs with adenine A, and cytosine C goes with guanine G, and vice versa. DNA is found in the nucleus. Due to its large molecular size, a messenger RNA strand, or mRNA, is needed to carry out protein information from the DNA to the cell's cytoplasm.
RNA, unlike DNA, is a single strand rather than a double helix. RNA is also made up of sugar, phosphate, and a nitrogenous base. However, the sugar is a ribose, and one of the nitrogenous bases are different from those of the DNA. DNA contains thymine, while RNA contains uracil. While RNA is transcribed by the non-coding strand, adenine is paired with uracil instead of thymine.
Let's get into the actual process of transcription. First, an enzyme, and if you don't know, an enzyme acts as a biological catalyst. Specifically, the RNA polymerase attaches to a specific section of the DNA called the promoter to begin transcription. In step 2, the RNA polymerase causes the DNA to unwind into two separate strands, a non-coding and coding strip. The non-coding strand is the one that reads or is being transcribed by the RNA polymerase. Since RNA is 5'3", the non-coding strand is 3'5", so that they can connect. That being said, the coding strand goes off on its own and is also 5'3", similar to the RNA strand.
In step 3, the RNA polymerase used the non-coding strand as a guide to pair bases one at a time, using the base pair rule to assemble free nucleotides into the complementary strand of RNA. A new RNA strand is created with the new bases A, U, G, C. Now here's a quick recap of the video. So step 1, transcription begins when an enzyme called RNA polymerase attaches to a segment of DNA. Step 2 is when RNA polymerase causes an area of the DNA helix to unwind and separate into two strands. The non-coding strand, which is read or transcribed by the messenger RNA, and this is the 3'5" strand, and the coding strand, which is the 5'3" strand.
Using the non-coding strand as a guide, RNA polymerase uses the base pair rule to assemble free nucleotides into a complementary strand of DNA. RNA transcribes the DNA non-coding strand and then binds it to a new nucleotide that is complementary, and a new RNA strand using the bases AUGC is formed.
Thank you for watching. Hope this helped.